So something that I've done recently that I haven't done for a while actually leads into this discussion, and it's around being away from Mary, my wife, and our brother Mina for a few months now, and it'll end up being about 6 months. You know, the care needs and the day-to-day life of you know, having a child that's so impacted is quite difficult, particularly around fatigue. And so Mary and Mina have gone overseas to be with Mary's parents for a while, and they're all sort of sharing nap times, and they're having naps at various times while Mina, you know, can't sleep during the night, and sharing that around a bit. So mine would be being away from them for such a long time, and unfortunately it's still going to be another couple of months, but they'll be back soon. All right, so thank you for having me here. My name's Steve Hille. My wife is Mary, and our little FOXG1 baby is Mina. I'm going to talk about our experience with FOXG1 syndrome and our foundation. So before I get into our story, I wanted to quickly talk about preconception screening. So anyone who is thinking about kids, you should be doing it. You should be telling everyone about it. There's really no excuse for not doing it anymore. When Mary and I, before we decided to have a baby, we went and met with the GP and spoke to him about it. And he sort of looked at us and said, well, you're not cousins, just go and get it done. And, you know, I really think that if we knew what we knew now, we would have gone down a different route and really tried to actually get the testing done. He really looked over it as if it was nothing, and talks I have with people since then, the attitude is still very, very similar. So please, everyone be doing that and talking about it. And there is a group called Mackenzie's Mission who are really behind this, and I would really suggest you also speak to and look those up as well, and they're mackenziesmission.org.au. So Mina was born in 2019 on my father's birthday, which was a really, really lovely joy for us. Mary had a fairly unremarkable pregnancy. She worked until she was about 8 months. We had all our regular checkups. Everything was fine for us. There was no abnormalities, no reason to be concerned. At Mina's birth, her measurements were perfect. There were really no issues, and we were really obviously happy and excited. She did cry more than we expected, but we were first-time parents, so it was difficult to know whether that was just us not being parents before, or if it was an actual issue. We really struggled with Mina's sleep. It was not uncommon for her to cry and scream and still be awake at 3, 4 in the morning, hoping to get her to sleep, and that unfortunately, for the large part, continues without medication. We went to a clinic at about 4 months to try and get us some help with her sleep, and they measured her up and realised that her head had barely grown since birth. So even though at birth her measurements were fine, it just didn't grow after that. So then we booked in to see a paediatrician, and the paediatrician held Mina and realised that she was very floppy. She didn't have much muscle tone, and she thought Mina was blind. Thankfully, at the moment, we don't think that's the case. Mina then went on to have a number of seizures, hence the epilepsy diagnosis. She's now a few months past 3. She has global developmental delay. She can't talk, she can't sit, she can't feed herself, she can't drink from a bottle, she can't crawl or hold anything in a meaningful way. So she's pretty well completely dependent on us. And this is quite typical of most FOXG1 kids. There are differences, but this is sort of about the average. So I was really lucky, and I think Mary was as well, to have really, really good parents. And you know, the experience that I think we looked at as being parents has become very different to our upbringings. You know, we both had very good parents, loving homes and so on. And I think in my mind, as I was growing up and wanting to become a father, I always thought that being a parent meant making yourself redundant, right, at some time, to your kids, so that you can pass everything you need onto them and they can live by themselves and you know, be productive and so on. And for us, that's not a reality at the moment, certainly without any sort of therapies. And on that, you know, our day-to-day lives are quite different. During my lunch break, when people are talking about their holidays and what's coming up and what movies they're watching, I'm on Zoom calls with researchers and you know, working on our data project and fundraising and all this kind of thing. And it also leads into an uncomfortable topic, which is thinking about your own mortality. You know, Mina requires, and a lot of the kids who have these sort of issues require so much care, and it's so frustrating and you're so fatigued that it can be quite difficult to think, well, who's going to provide this level of care and this level of love once we're gone? And that's, you know, that's I guess, why we're here and why we're trying so hard with the researchers, to try and get that sorted out. So when we found out about Mina and her issues with the paediatrician, that was right around the time the government made whole exome sequencing available on Medicare. So to the best of my knowledge, it's still the case. Any kid under 7 with an undiagnosed medical condition gets whole exome sequencing for free. We went and did that. And a quick side note on that, we're hoping that they will change us to whole genome sequencing as opposed to whole exome sequencing, so they're not missing out on any mutations that we might miss with just regular whole exome. The results came back and we learned that Mina has FOXG1 syndrome. Never heard of FOXG1 before, I had no idea what it meant. And it had been inherited to her by Mary, which is extremely rare, especially for FOXG1 syndrome. This is known as mosaicism, and we don't see it very much at all. Mary has a small amount of cells affected in her body, but she's asymptomatic. She's perfectly healthy. So since then, we've been getting her the support she needs with physio, OT, speech therapy, and so on. And so, don't throw any tomatoes at me, but the NDIS for us have been flawless. We've had not a problem at all. We've been really, really lucky and very fortunate to be in Australia where we have such good governmental assistance. So following a diagnosis, I looked to get involved, and through a series of events, ended up forming the FOXG1 Research Foundation in Australia, and that's as part of the Global FOXG1 Research Foundation, which is very well established and has achieved some really incredible things in a short amount of time. The mission of the foundation is to find and fund world-leading scientists to understand as much as we can about FOXG1 syndrome to lead to therapies and ultimately a cure. The foundation, we have a path to a cure, and is made up of many parts, including the patient registry, a natural history study, a biobank for skin samples that allow us to grow induced pluripotent stem cells for our research. We have, and we're doing those per exact mutation for the research. We have our own FOXG1 Centre of Excellence in New York that is run by the Lees, who are both geneticists or genetic professors and have a daughter with FOXG1 syndrome named Una. We're funding some really unbelievably clever folks all around the world. We know of about 1,000 people worldwide that are affected by FOXG1 syndrome, including about 20, maybe a few more here in Australia. But that number is really wildly low for a number of reasons. One of them is that testing hasn't really been available. And especially in a third world, we think the number will be significantly higher where the testing is out of reach, either practically or financially. And even for us, FOXG1 syndrome used to be incorrectly categorised, and sometimes still is, as Rett syndrome. And Rett is the MECP2 gene that affects Rett folks, and for us it's just a FOXG1 gene. This was identified in about 2008 and broken out. So FOXG1 is a single and incredibly important gene, particularly during fetal development. What we understand, it's the orchestration gene that tells other genes when to turn on and off. And while you're being created, it's a very important thing. It used to be called brain factor 1 as well. So we're excited about the prospects, particularly on gene editing. All of you in this room have FOXG1 genes in your body that are expressing proteins. And because of that, for the most part, we're all living healthy, able lives. We think that if we can fix a mutation, and obviously every mutation is a bit different, and there are different types of mutations and therapies for one type of mutation won't work for another, and so on. But we think if we can fix that, then over life we'll see a slow and steady increase in capabilities, just because those proteins are being created correctly and all the follow-on effects from that. Within Australia, we have a really incredible board of folks and volunteers. We're actually working on a data project at the moment where globally we're trying to collect all the data, make it central, and bring the researchers to the data with tools that are much simpler than what they use at the moment. At the moment, it's really a lot of hand-cranked, command line tools to find answers to their questions, and they're not really repeatable, we're finding, and so on. So there's lots of platforms that are popping up now, and we're looking into one of those right now to try and make it a bit easier for them. Let the geneticists do the genetic research, and we're getting some bioinformatics folks to help us answer their questions from a technical perspective. We're really lucky to have Dr. Fabian Delarue from Macquarie University as our sort of partner in research, who amongst other things is dedicated to FOXG1 syndrome. He's finished one PhD in FOXG1 syndrome, and I had a call from him about 2 hours ago that it looks like we've just got another one. So that's unbelievably exciting for us, and we're really, really happy for that. He's working on a number of things, brain organoids, Prime editing is a really, really exciting one for us. We think that's probably going to be, well, I certainly think it's probably going to be one of the biggest chances we've got of it, well, in our case, Mina has a mutation where her C and G is swapped, one C and G. And so, as an example, prime editing is able to swap single pairs over, and we think that will be really the big one. We believe that as the research goes, unlocking more and more information, as possible, will contribute to the science and a huge amount of other genetic-related illnesses. For example, we could have a therapy, but if we can't get through the blood-brain barrier, it's not really that interesting, right? There's lots of other things that have to come into it. So everyone's sharing, learning from each other, and that's really good. We need about 30,000 per PhD candidate per year, which is about the average for most universities. So anyone who's looking to try and stand up some research for your own mutations or mutations that affect you, it tends to be about 30 grand a year. And obviously we want to run as many as we can. So anyone who is interested in partnering with us, we're always looking for biofarmers, more researchers. We're looking for anyone to share or obtain information from. Please come find me on the internet, however you want to do it. Really love to catch up with you. Thank you for all being here, and thanks for listening to me.